Welcome to MethMotif 2024, our first update to the integrative cell-specific database that joins transcription factor binding motifs with DNA methylation. The MethMotif project is a collaborative project spanning multiple universities and countries. The database update offers two new species, mouse and plant. For the human portion of the database, we have also expanded to 655 transcription factor binding sites across 16 cell lines, encompassing 2,473 ChIP-seqs and 22 whole genome bisulfite sequencing experiments. Starting at the search bar, users can query the database by entering a transcription factor of interest. As the name enters the search bar, MethMotif will return transcription factors matching that as a drop-down list. Let's choose CPP beta. After selecting a transcription factor, click on the search icon to launch the query. Upon completing the query, we land on the Motif search page. MethMotif has provided all the results for CPP beta within the human database. We can notice this includes 8 different cell types. At a glance, we can see the global CPP beta motif within various tissue types, from lung to liver. Another essential feature of the Motif search page is the star present after CPP beta. This star denotes the pioneer status of the transcription factor. The pioneer status is something we will expand upon a little later. Let us now click on CPP beta within the K562 cell line. After clicking on the motif, MethMotif presents us with the motif card. The motif card contains all the relevant information for the chosen motif. We will start with the motif logo. We can observe the conserved ATTG motif that is typical of CPP beta. Hovering the motif will increase its size for better viewing. Below the legend, we can modify the motif displayed. These modifications range from changing the methylation level to changing the entire plot from an entropy logo to a frequency logo. Scrolling further down, we're greeted to 5 columns. The first 2 columns provide basic details about the cell line and transcription factor. The third column covers information about the motif, such as the motif location. By scrolling down to the motif location and clicking on the pie graph, we can increase its size for better viewing. Skipping the 4th column for a moment, we can view the 5th column, the pioneer factor information. Here we find the evidence to support the previous pioneer factor claim. The evidence is presented in a manually curated manner. For each characteristic, we observe a true or false determination along with a PubMed reference for that determination. These characteristics culminate in a true or false statement about the pioneer status of the current transcription factor, as well as a reference. Moving to the 4th and final column, we can display the true power of MethMotif in this latest update. In this column, we segregate CEBP beta with each of its most common cofactors. Scrolling down to the cofactor report and clicking on the eye icon will reveal to us the cofactor report. Within this report, we can view the motifs from the regions shared between CPP beta and each of its cofactors. Along with updated logos, we can see the methylation directly within the motif, as well as plus or minus 100 base pairs around the motif region. Finally, we also provide read enrichment. If we were to hover down to CPP beta and CPP delta, we notice that the read enrichment is surprisingly higher than other partners. Closing out of the cofactor report and selecting a specific cofactor from the dropdown will let us drill down into this data. Clicking on ATF4, we can see the global motif contrasted against the overlapped motif. Below the motifs are the relevant methylation heat maps for within the motif itself, as well as within the 100 plus or minus base pairs. In the next tab, we provide the updated genomic location information specific to the current cofactor in an interactive donut plot. Moving to the final tab, we provide an interactive gene ontology plot calculated on the cofactor-specific loci. In this Go plot, 3 gene categories are available within the drop-down menu. This concludes the MethMotif 2024 database walkthrough.